Welcome to the first remote lab. COVID has definitely promoted us to start moving towards these, to start learning how to convey information that would normally be learned in a lab environment as well as we possibly can when doing it remotely. Microscope labs usually come with a lot of issues, especially because there's a lot of sharing instruments. Microscopes themselves can be very expensive, and people can break glass slides, and then there's glass shards, so it's a lot. Let's try to cut down on all the mess, but keep the learning experience as best as we can. If you are able to get your hands on a microscope, there's a few things you need to know first. There are cheaper microscopes on the market, ones that you can even hook up to your laptop via USB. They're very accessible to us students. However, we're talking a huge range of qualities and prices though, so make sure you do your research before buying one. One time I got a little $20 microscope all excited and it did not work at all. So what I'm saying is borrow one from an actual lab or professor if you can. So if you do get one and it's a good heavy one, carry it with one hand on the arm and one on the base. There's so many bad things that can happen if it drops or breaks in your hand due to its own weight. And they're also so expensive, not to mention if it drops on your foot, it's going to really hurt. Or if it even drops on someone else's foot, it's going to really, really hurt because then you got to say sorry, right? That sucks. Once you've got the microscope in place and plug it in, place your prepared slide. We'll get on how to do that later, don't worry, on the stage, which is aptly named. Now, you see these cylinders? These are your lenses, the objective lenses, and the body tube connects them to the eyepiece where you look into. You'll want to start with the lowest power or smallest objective lens, which magnification will depend on the type of microscope you have, but we start with the lowest power magnification because it's a lot easier to find what you're looking for from further away. If you're wanting to see the chloroplasts in a cell and the spongy mesophyll of a leaf sample, you're going to have to find the spongy mesophyll first, right? Increase magnification until you find the general area and close-upness you want, and then we're going to bring that image into focus. The focus knobs move the body tube, and in turn the objective lenses further or closer to your sample. Start with the larger one, creating larger movements, and once it's pretty clear, you'll get even clearer with the fine focus knob, which gives you precise control over the clarity of the image. Keep fiddling around until you get what you're looking for, but be careful. If you lower the lens too much, too close to the slide, you can actually break the slide if the lens pushes through it. So just keep yourself aware how close the lens is. Again, you don't want one, broken shards of glass, and two, to have to re-prepare your slide. So how do you prepare, or what we call mount, your slide? There's 100 different protocols and 100 different ways to do this. I'm going to give you the general methods I'm most familiar with and have gotten good results with. But just keep in mind, if you have a protocol or instructions already, forget these and use that. Again, this is just if you're completely winging it, you've got a general idea. So first, you'll harvest yourself a thin slice of the tissue and place it on the slide. Try not to use your bare fingers. Use a clean knife and tweezers or fresh nitrile gloves. You don't want to contaminate anything with your own skin cells or anything else that's on your hands. You don't want to be looking at a plant and be like, oh my God, these are animal cells. So mostly you'll be doing wet mounts. So this requires a drop of water on top of the specimen. Just one drop, don't get too crazy. You don't want to wash it away. So perfect drop, a little bead of water like you see in this graphic. Now, your cover slip is a very thin piece of glass or sometimes plastic, and you want to place one side of the slip just on the edge of the water drop, like in the graphic. Slowly and carefully, to prevent air bubbles, lay the slip over the drop and it should adhere perfectly to the slide. Now, there are times when you'll want to use a stain, which is a dye that colors only specific parts of your specimen to make things much easier to identify. You'll see that in this lab. There's a bunch of different colors that you'll see. There are lots of stains available, with each having their own purpose and own instructions to go with it. But there's two basic ways to stain. For bacteria mostly, but you can do it with plants, you air dry and then fix the specimen to the slide with extreme heat, then stain the dried specimen, refraining from adding a cover slip. The other method us plant people will be using most often is still with that wet mount method. After we place the cover slip on top of the drop of water, we place a drop of stain on one side of it, touching the water under the slip. Then place a paper towel on the other side. This paper towel will begin sucking up the water, and the dye will displace the removed water under the slide. Science. What you'll usually see first in any microscope lab involving plants is this, the classic onion root tip slide. This is the location of meristematic activity, cell division. Specifically here, mitosis or nuclear division, as in the nucleus where DNA is located, not like nuclear power. There are 5 stages of mitosis that you can see under the microscope, and they're pretty distinctive. If you see these different stages and these dark, clear chromosomes, it's a good guess you found a meristem. Those 5 stages you'll see are the interphase, where the cell goes about its normal processes. At the tail end of interphase, DNA is replicated, so there's 2 of everything moving into prophase, where we see chromosomes gathering and condensing. Remember, 2 of each. Metaphase, where these chromosomes are brought to the center of the cell. Anaphase, where the pairs are split and each half is pulled to an opposite side. And telophase, when they arrive to these opposite sides. This is a very simplified explanation. Please look into it from a more in-depth source if you need more information. Chromosomes and chromatids in particular get a little messy, and it's good to have someone that has a very definite way to explain them so you don't get confused. But I'm not going to do that today because I want to get on to microscope stuff. So we have our normal, typical plant cell, and under the microscope, it looks like this. The stain used by this slide makes our cell walls stand out really well, as does the nucleus. The vacuole, however, is very hard to see. It's just kind of light or white, really. So as you can see here, this is a good example of different stains doing different things and staining different things for different purposes. Luckily, this is a remote lab, and we can take advantage of technology and show this image of a scanning electron micrograph. It's taken by a scanning electron microscope, which creates really amazing, clear pictures very close up. If you are ever having trouble while looking up micro anything, try searching for scanning electron micrographs of your subject. Just look here, so much more in focus than a normal microscope image, and you can see organelles that weren't even discernible in the previous picture. That Golgi apparatus is still pretty hard to see, but whatever. Someone could point it out, and that's what matters. But scanning electron micrographs can't show you everything a normal microscope can either. These are chloroplasts moving around in real time thanks to the movement of cytoplasm called cytoplasmic streaming in the cell. If you want to see this yourself, track down a plant from the genus Elodea. They're aquatic and very common in plant microscope labs for the reason we see here. The leaves are thin enough we can put them straight on a slide and still see what's going on inside with ease while they're alive. All the other organelles in this image are clear, however, without stain, and so it's hard to see anything else except the chloroplast and cell wall. Different purposes, different stains. There's just so many protocols. Again, these are bare basics. All right, let's move on to our basic cell types. Parenchyma cells are generic, pretty much what we've been looking at for those past 3 images. Thin cell wall, nucleus hanging out here, and our large intercellular spaces. In Collenchyma cells, we see that thicker, uneven cell wall at the edge of those parenchyma cells. Less intercellular space, and despite this being a cross-section, we know that these cells are typically more elongated than parenchyma cells because we're smart. There should be nuclei in these, but it's a little hard to see in this image, so take my word for it instead. If I had to guess, I'd say these Collenchyma are just under an epidermis, since we see a lot of space and possibly trichomes above, if you see that. Whoa, massive cell walls. Here's our Sclerenchyma, dead at maturity, thick cell walls. These are fibers in a yucca plant, strong and sturdy enough to support the plant and also be used as textiles, and they have the lignin in those walls to thank for that. Using a less high power objective lens, remember that means we're less magnified, more zoomed out, we'll take a look at the vascular bundle. This image is a little misleading because of this concave curve, but that space is the outside of the stem. So we have the phloem closest to the outer edge and the xylem further in, because we want our water protected more than our food. These vascular bundles sit in and are supported by ground tissue in the stem and have that distinctive vascular cambium bisecting the 2 tissue types. Now, we do have 2 different organizations of vascular bundle in plants, the haphazard anywhere goes method of monocots and the ring structure of dicots. Now, it's hard to tell when looking so close up, but what do you think on this image, dicot or monocot? Yes, dicot. Not only because we have that edge right nearby, but because of the vascular cambium. Look again and see monocots don't have vascular cambiums. Their vascular bundles are all kind of shoved together, phloem and xylem, right next to each other. Onto epidermal tissue and some very pretty images. These images both show the pavement cells, which make up the bulk of the epidermis, but the stomata are much easier to see in the rightmost image. These two bean-shaped cells that make the little kiss faces are the guard cells, with the stomata in the middle, these pores that let gas in and out of the plant. You can pick them out in the left picture, they're just not as neat, kind of harder to tell apart from the pavement cells. So if you're looking for guard cells in any of your own specimens, keep in mind you might have to look really close and make a few educated guesses. Speaking of epidermis cells, we have our root hairs and trichomes next. Root hairs are almost always, in my experience, single-celled. You'll typically see the cell in the epidermis, and coming out of it is the long hair which uptakes water and nutrients for the plant. Trichomes, however, are above ground, and they can be multicellular. Notice those black lines and the really dark nuclei in each cell that makes up the trichome. So if you're having trouble identifying which is which, if someone just hands you a slide and doesn't tell you root or shoot, keep an eye out for stomata like you see in the background on the trichome image and multicellularity. They also come in many shapes and sizes, while root hairs typically all look the same. If you don't see stomata and it's a single cell, you can make that educated guess it's a root hair, that or someone's being a jerk and trying to trick you. Periderm or bark tissue. We see that epidermis now sloughing off, giving way to the cork cells created by the cork cambium. These are dead at maturity and hollow, basically just cell walls full of lignin and suberin. Remember, lignin is rigidity and suberin is waterproofing. Here we have a leaf now with the epidermis on both top and bottom protected by the waxy cuticle. Palisade mesophyll is densely packed together and each cell is densely packed with chloroplasts. Below is spongy mesophyll, light and fluffy, to allow gas exchange to the palisade layer, which needs a lot of it to keep up such high rates of photosynthesization. If you squint hard, you can kind of see some possible guard cells here on the underside, but nothing is obvious, so I don't want to make any for sure statements. This cluster of blue cells to the left here might be a vascular bundle, but it could also be some mangled tissue where someone got too excited with a knife. Hard to tell just from the edge, not the pretty specimen, but I want you to be aware that not every slide will be perfect, and there is some doubt when you're looking at these images, especially if your stain gets a little messy, if you didn't mount it quite right, and again, if you have some contamination. So again, these slides you find on the internet, usually pretty good, but if you're a first-time slide mounter, again, you might have some trouble identifying things, and that's okay. Practice makes perfect. Here, however, are some very nice specimens. On the left, we have a fruit with the ovule inside the locule, simply the chamber the ovule is in, and then the pericarp surrounding the locule. As nice as this image is, I personally was a little confused with their labeling of ovary wall pericarp, and assume you may be too. The pericarp is the swollen ovary wall tissue, and not any one part of this fruit surrounding the locule. So when they say ovary wall pericarp, I assume they just mean this is the pericarp made from the ovary wall or something. On the right, we see the male part of the flower, the anther. This is the housing for all pollen, and it's borne on the filament, which is just the stalk. Speaking of pollen, pollen close-up is amazing. There are so many diverse types of pollen, all with shapes specific to their methods of dispersal and pollination. Typical microscopes give us a view that's cool, yeah, but check out scanning electron micrographs. Pollen is its own art. Look at all these different patterns and spikes, and yeah, just pollen's cool. So if you ever get a chance to check out a book that or an internet source that has a bunch of scanning electron micrographs of pollen, check it out and look at the species that each belongs to. Try to figure out why do they have this shape. If this is spiky, is it meant to cling on to something better? If it's very light and has many holes in it, maybe it's meant for wind dispersal. So it's kind of a cool activity to just have fun with yourself. But after that, we're done. Very short lab. We're not going to take 2 hours to do a lab just to draw it out, you know? It's just easy, simple, and I will see you guys later.